So this is the rigor mortis modeling by Nabil and Jan. So first we're going to describe the myosin and actin. So this is the parts of the myosin. It involves the myosin heads, which have ADP and ATP attached. And this is the actin, troponin, and tropomyosin. At rest, the adenine diphosphate and phosphate molecules are here with the myosin. And calcium is released, which are these red beads, from the sarcoplasmic reticulum, and which then binds with the troponin molecules, which causes the tropomyosin molecules to move and attaches to reveal on the myofilaments. The single phosphates on the heads get released, and they reach for the active sites, forming a cross bridge between the myosin and the actin. The adenine diphosphate molecules is then used during the power stroke when the myosin heads pull the actin inwards, like this. And once the ATP is spent, ATP is attached to the myosin heads. So it becomes that. This triggers a release from the myosin heads and the active sites, which breaks the myosin and the actin apart. This breaks down the ATP to ADP and phosphate, which returns back to these two. Myosin heads move back to resting position, which is called the recovery stroke. If there is calcium, it will repeat. Sarcomere will shorten until calcium ions are back into the sarcoplasmic reticulum. So now we're going to talk about the rigor mortis. So the rigor mortis is also called the stiffness of death. After 3 to 6 hours of death, it loses its energy source, which is ATP. And the organelles are then leaking calcium, which is then continues the cycle between the myosin and the actin, which creates uncontrolled tensing stiffness of muscles. And this is going to be stuck in this position from 24 to 48 hours. Without the energy, ATP, the myosin heads have no trigger to release, so it stays like this.